All right, hello everyone, and welcome to Therapeutic Approaches for Rare and Nonsense Mutations. I'm really excited to be back at ResearchCon for our second year of offering this event. Those of you who attended last year may remember me from the careers panel. I am Zan Novakovsky. I'm an assistant professor at Florida State University College of Medicine at our campus in Orlando, Florida. I am 36 years old and living with CF that is caused by very, very rare genetic mutations. So I am really excited to have with us tonight Dr. Phil Thomas of the Cystic Fibrosis Foundation and various academic units, and also Dr. Pete Hagge from Emily's Entourage, here to talk with us about some of the latest and greatest innovations in therapeutics for rare and nonsense CFTR mutations. So before we get started with our content tonight, I want to do just a little bit of housekeeping for y'all as your session moderator, and also let y'all know that I will be running the chat for the event as well as the questions and answers. So first and foremost, I want to go over a few other items related to technology and how we can interact with each other. We'll be taking audience questions during the session. So if you have a question, you can submit it at any time by clicking the icon on the right side of your screen labeled Q&A. You can also see other questions there and give a thumbs up if you want other questions to be answered. I'm going to try to aggregate the questions that we actually address verbally. So we're going to tackle very general topics, but I will also try to respond to you inline in the event chat to make sure every person gets some individual feedback. It's important to note that this is not a session where you can receive direct medical advice. If you have a question about your own care, you should absolutely follow up on that with your team. But we do encourage you to think creatively about how the content from this session may impact the kinds of questions that you want to ask your team and the kinds of dialogues that you want to have. We also definitely want you to get even more fired up about participating in research, and especially those of you who are living with rare and nonsense mutations yourselves. That's a unique superpower in helping the science advance to make sure that every single person with CF can receive the most person-centered care that's going to help you have your best health for a very long time. So we definitely encourage you not only to ask good questions tonight about how to get involved in research specific to your circumstances, but also to get involved with some of the big research activities nationwide, including Community Voice and Research Voice with the Cystic Fibrosis Foundation. If you're interested in learning more about ways to contribute data to CFF, definitely connect with our CFF staff at the event and also go on the program webpage, or you can creep on my Facebook and slide into my DMs and I'll help you out. Finally, how to get to chat. You will notice the chat bubble icon on the right side of your screen. That is different from Q&A. If you want to ask a general question to our panelists, use Q&A. If you want to talk with other people who are attending the event, use chat. And if you have any tech problems during the panel, you're going to want to navigate back into the general session area and go to your tech and program support tab. Those folks are going to help you out with whatever you need. You can also, if you're having trouble reaching tech and program support, just send a message in the chat and I'll make sure that you can connect with that. With that, I want to give our other speakers a chance to introduce themselves, and then we're going to start out with some content introducing basic concepts and terms. Phil, why don't you go ahead and start?
Yeah, sorry, I only have one screen here. I have to get back to where I was.
No worries. So just so y'all know how this is going to work, Phil is going to be sharing.
No, I forgot to put the share screen thing first. So
that's
sorry.
Okay. We don't have to do that yet. We want to see your faces while you introduce yourselves. Why don't you share the slides after we do introductions?
I can't do that.
All right. Well, you know what we look like now. So Phil and Peter, go ahead and introduce yourselves, and then we'll get into the slides.
Okay, I'll start while Phil's getting the slides up. My name is Pete Hagge. I'm an associate professor at UCSF. My primary research focus is trying to develop small molecule paradigms that could be therapeutically efficacious for cystic fibrosis. In addition to working at UCSF, I'm also the scientific director at Emily's Entourage, which is a charity focused on development of therapies for CF caused by nonsense mutations.
Hey, this is Phil Thomas. The one reason they had me on here is they thought I could handle the slides, and we could see that didn't work out so well. But in my other job, I'm a professor at the University of Texas Medical School, which is located in Dallas. I've been working on CF for almost 30 years in that laboratory. In my other duties, I'm the senior scientific advisor to the CFF laboratory that's up in Lexington, Massachusetts. I go up there monthly, at least I did before the pandemic. And I'm also a founder of two biopharmaceutical companies that operate in the CF space.
Excellent. All right. So we're going to just get right into our presentation after a brief disclosure. Pete and I had nothing to disclose in terms of conflicts of interest. Phil did have a couple of disclosures. And again, these presentations are going to be recorded. So if you want to view anything in detail, definitely connect with our virtual events account at the CFF or connect with one of our staff in the chat, and they'll help you understand how to access those recordings post-event. But let's get right into the scientific content to make sure we can stay on pace because we got a lot to cover tonight. So let's start out with the flow of genetic information in the whole process associated with the CFTR protein. We're going to go over some basic concepts and terms, and then we're going to orient y'all to some emerging therapies and the science behind them.
Yeah, so just to follow on from Zan, we are going to start with some basic concepts, and we'll hopefully try and explain why some of the therapies that are available now might be more applicable to some of the mutations that aren't amenable to therapies, and then explain some of the concepts about how some of the newer therapies that are in development may affect other processes in the cell, but also therefore be of benefit from CF. So in this first slide, we just want to refresh everyone on the basic concept of how information flows from the genes to a protein. So DNA is a molecule that's found within the nucleus of cells, and DNA encodes genetic information in segments that are called genes. The CFTR is found in the CFTR gene, in DNA, in the nucleus. For it to become a functioning protein at the far end of the slide, the far right side of the slide, it goes through several intermediary steps. The first is production of another molecule, which is also linear, called RNA. This is essentially a copy of the DNA, but it's more transferable in that it's out in the side of the cell so it can be looked at by other machines within the cell. The first machine that looks at it is the ribosome. So this is a large, complex machine formed of proteins and, again, more nucleic acids, which chugs along the RNA, translating the genetic information into protein information in the form of a nascent polypeptide, in this case CFTR. Originally, that protein is kind of partially folded. It doesn't have its native status, its native form. And through a process called maturation, it does gain that native, more compact form, which means it's permissible to be presented at the cell surface. And so, for instance, in the lungs, when it gets into the cell surface, that's where CFTR works as the protein. It conducts chloride. So the main concept here is that there are multiple steps in the pathway from the gene in the nucleus to the protein at the cell surface. Sorry.
So Pete's going to hand it over to me. I'm kind of a simple-minded person, so I hark back to my childhood in South Dakota, and we're going to use cattle as to indicate chloride ions. And in CF, the problem is we can't move the cattle from one pasture to the other. We can't move the chloride ions across the apical membrane of the secretory epithelium, which is required for control of fluid secretion. And as scientists, we like numbers. So the number we're really interested in I, and that stands for the current, or in other words, the amount of chloride ions that pass across the membrane per unit time. And that number I is determined by three other numbers, and that's the number of gates or CFTR molecules that are in the membrane or in the fence, how wide those gates are open, and the fraction of time that they're open. As you can imagine, if you have more gates, you can move cattle more quickly. If the gates are open more widely, you can move them more quickly. If the gates are open rather than closed, you can move them more quickly. In CF, there are alterations in the gene sequence, as Pete described, that prevent this process from occurring properly. And one way this can happen is that the gates don't form properly and they never get put into the fence. There's no gate in the fence. There's a number of ways that can happen. Another way that can lead to the disease, if the gates are there, but they don't open widely enough. And so that would be a measure of PO, or the open probability. Now, luckily, we have small molecule modulators that act on CFTR to, one, increase the number of gates that make it into the fence, or the number of CFTR molecules that make it into the membrane, and or potentiators such as ivacaftor that control how wide the gates are open. Pete's going to now explain this in a more kind of scientific nomenclature.
Okay, yeah, so we started off with an analogy of what's going on with cystic fibrosis in that cows can't get from one pasture to another. And we've also used the concept that what's causing this is mutations in the CFTR gene. So in the nucleus, in the DNA, there are mutations. And what are they? And what are rare mutations, nonsense mutations, and conversely, what are common mutations? Well, if you look at the CFTR gene and sequence it, you can find where the fault or the mutation is within that gene. And if we simply consider how frequently that mutation is seen in subjects living with CF, we can build up a frequency and consequently calculate a percentage of the population that have that mutation. And simply put, the more often a mutation is seen, the more common that mutation is. And the less frequently that mutation is seen, the less common it is, the more rare it is. And essentially, that's what a rare mutation is, a mutation that is just not seen very frequently in the CF population. There's real data from CF subjects shown on this slide, and as you can see, the top mutation, which we're all familiar with, F508del, delta F508, is extremely common in the CF population, as we all know. It's seen, in a far more dominant percentage of subjects than any other mutation. It's the most common mutation. As we go down that table of different mutations, we see that their frequency drops, they become less common. But what's interesting is the second mutation there is G542X. That's a nonsense mutation. The next mutation, G551D, is again a missense mutation. You keep going down, there are different types of mutations. So what's clear is that all rare mutations are not nonsense mutations. And in fact, nonsense mutations are not necessarily always that rare. What's common about them, though, is that rare and nonsense mutations aren't currently necessarily amenable to therapies. There are some exceptions to that. As you know, F508del has Trikafta, G551D has Kalydeco, and some rare mutations we've actually found, and importantly found, that they're amenable to therapies as well. So that's the distinction between what a common mutation is, a rare mutation, and a nonsense mutation. We'll expand on some of these things, particularly nonsense mutations, in the coming slides. Okay, so what are these mutations? So what are these different? So what do these different mutations actually do to the CFTR protein? What we've actually done is en masse classified all of the different type of mutations into similar families. So again, if we start with the normal situation, which is shown on the left-hand side, there's the flow of genetic information from DNA to RNA, the protein being generated, inserted in the apical surface where the normal CFTR functions as a chloride channel. The first class of mutations is nonsense mutations. Here, the mutation means that an amino acid is not encoded within the protein. So instead of producing the full-length CFTR protein, a shortened form of that protein is generated. And as we know, and I've already seen one question, the nonsense mutation or the X mutation, the premature termination codon, they can occur at different locations within the CFTR protein and produce different amounts of CFTR protein. Currently, it's believed that the longer the protein that's generated, the more likely it is to be close to normal CFTR in its function. But that's something that is an evolving question, and we'll have to find out more There are therapies, and again, we'll get to this, that are in development where it doesn't really seem to matter where the X mutation is found. The second class of mutation, class 2, the protein stops it being able to fold appropriately, so it doesn't mature appropriately and it can't be inserted in the cell surface. An example of that kind of mutation is F508del. The third class of mutation is where the protein is formed in its complete length. It does mature, it does present at the cell surface. However, it doesn't work there. Currently, we have modulator therapies that are amenable to class 3 mutations. So a classic example of a class 3 mutation is G551D, and potentiators such as Kalydeco are used for that. A classic example of a class 2 mutation is F508del. We know that correctors in conjunction with potentiators are used for these mutations. But the take-home message from this slide is that there are multiple mutations, and those distinct mutations can cause all different types of defects in the CFTR protein. So it's not necessarily the case that one type of therapy is going to fit all of the problems, okay? And that's why so many shots at goal have been taken in terms of developing CF therapeutic approaches. So if we go back to the cow analogy, we can contemplate these different mutations in the simple form that Phil suggested, using cows, gates, and fences. So 508del, F508del, that's the case where the gates aren't put in the fence appropriately. G551D, the gates are put in the fence, but they're not opening appropriately. So again, in both of those instances, the cows can't get through the gate. With a different type of rare mutation, R347P, here the gates are present, but they're kind of narrow. The pore is constricted. And we can also conceptualize how different types of modulators work. So for instance, again, with G551D, Kalydeco is a potentiator. So what's it doing? It's ostensibly opening the gate that's already in the fence and allowing the cows to come through. So we've talked about broad concepts about what mutations are and how they affect the CFTR protein, and shown that there are multiple different ways that the CFTR protein can be affected. But what I'm doing in this slide is showing you exactly what the CFTR protein does in the cell surface of the lungs. So the schematic at the top left is what you might imagine we see if we use a really fine magnifying glass to look at the lung surface. The lung surface itself is coated in fine hair-like projections called cilia. Those cilia are bathed in a fluid, an aqueous fluid called the airway surface liquid, and that fluid supports the cilia. Interacting with the tips of the cilia is the mucus, which is the yellow blobs. And so in a healthy lung, at the top there, a healthy lung, the mucus interacts and the cilia beat in a regular manner, in a pulsatile manner, which moves mucus up through the lungs and out of the lungs. The mucus binds pathogens like bacteria, viruses, and also particles, and so that's a primary mechanism of defense that the lung uses. What's controlling the airway surface liquid, the fluid that bathes the cilia? Well, it's CFTR function in part, and so when CFTR functions appropriately, the ASL is appropriate, the cilia can work, the mucus can be moved. Let's contrast that now with cystic fibrosis. Here, there's no CFTR function, so no chloride. That means the cilia, can we go back a slide, sorry. That means the cilia aren't bathed appropriately, they collapse. They can't move the mucus anymore in their concerted, coordinated manner. That means mucus builds up, and more so because there is less hydration at the lung surface, the mucus is more sticky. That allows bacterial infections and viral infections to seed more effectively and propagate more effectively, which is what causes the infection and the inflammation. Okay, next slide. Okay, so just to drill and reinforce in on those concepts of what's going on with available modulators. With a potentiator, the CFTR protein is at the cell surface, but it doesn't work properly. When it binds the potentiator, for instance, Kalydeco, then it allows the protein to work effectively. pump chloride out of the cell to hydrate the lung surface and allow the lung to work appropriately. In the case of correctors found in in drugs like Trikafta, there are multiple defects in the protein. It's partially folded, immaturely folded, so the small molecule modulators bind to increase the folding and maturation of that protein, allowing it to be presented at the cell surface. But then another type of modulator, the potentiator, is needed to allow that protein to function appropriately. So what we've covered today is the mutations, the distinct types of mutations, the fact that they can affect CFTR protein differently and distinctly, and what's available with therapies that we have so far.
Okay, I'm going to pick up the theme here that Bill Gatch mentioned in the plenary earlier about efforts at the CFF lab in Lexington, as well as Bob Bridges' lab in Chicago and my own laboratory in Dallas to test over 650 different disease-associated variants or mutations. And what I want to do is I just want to come back to the theme of the cattle, because all we're really interested in here is I, the rate at which the chloride ion can move across the membrane, and is that appropriate for normal physiological function in the case of CF not. But what the real question is what's going to happen if we treat these different rare variants with the approved therapies. And so I know it seems incredibly daunting to think about, say, 650 or 800 or 900 different CF causing variants. But what I want to go back to the analogy is there are a lot of commonalities between what the ways in which these different variants prevent CFTR from functioning properly. In other words, there are variants that prevent the maturation, putting the gates in the membrane where they can be there. There are other variants that affect the ability of the gate to open, such as G551D. That's, at least we know in the case of G551D, that's responsive to Kalydeco. And there are other variants that do other things, such as affect the width of the gate, or how much rate of chloride ion can be through that particular channel. So to try and identify these commonalities, we did something that scientists just call an empirical approach. And what it really means is we just have to have a way to measure I in a convenient way very rapidly. And when we measure I, we really don't care if I is low because N is low, or I is low because PO is low, or I is low because gamma is low, or some combination thereof. What we really care about when I is low, if we put Kalydeco on that variant in that system, does I increase to a point where we think this might have therapeutic value? So that is the basis of a so-called theratyping. In other words, we're trying to group different variants into their responsiveness to a therapy. And we don't really need to look at this flowchart in too much detail. Just should suffice to say that we're in consult with the CFTR2 group led by Gary Cutting at Johns Hopkins University, and they're providing us all the molecular genetic data on these variants. And as an aside, I will say, as some of the variants they have provided to us, we find out that they actually do have functional CFTR. In other words, that variation is probably not disease-causing, and that's important information to know for the patients that harbor that particular variant, because that's probably a benign change and there's something else that's wrong. And that information is critical for getting people on drugs that they'll respond to. So we just have ways that we introduce these variants into DNA, the bacteria, we amplify these, and we express them in a mammalian cell on a tissue culture plate, and we make an epithelium on a support on a membrane, and then we can measure chloride movement across this membrane. And this just shows like how careful we are to make sure that we get good good data, because we don't want to mess up this process by providing bad data, because that makes people doubt the answers that we get. So I'm just going to show you here on this next slide a few of the kind of examples, and I think there were 400 or so different variants here on this plot on the bottom. And let's focus on the part of the plot that is in the circle, right? The bottom line is, in the black is the amount of CFTR function without any drug treatment. And you can see that these tend to be fairly low, but then you can see if we look all the way to the right-hand side, there's a number of them that have quite a bit of function, and those are the ones that are probably not causing disease. But let's look at the ones in the middle, and the various colors indicate treatment with various approved therapies. And what you can see is there are some of these variants that respond very, very well to the therapies. In fact, returning function that's even greater than wild type. Conversely, if we look far to the left-hand side, you can see there's quite a few variants down there that don't function well and do not respond. So the bottom line is what we're hoping to do is identify additional rare variants. What I should say is some of these variants, there's only one example of out of the almost 70,000 patients that have been examined so far. So that's as rare as it gets. It's 1 out of 70,000. Okay, I'm going to hand it back to Pete now, and he's going to talk about new therapies that don't rely on the approved drugs.
Okay, so to recap where we are, we've covered available therapies and considered theratyping, which is a really large effort to see whether or not the efforts that have been put into developing drugs can be applied on a broader sense to other mutations. And there is precedent for that. For instance, when ivacaftor was originally developed, it was developed for one mutation and it's been expanded. So we know, or we predict at least, that this theratyping effort will chip away at the 10% of CF subjects that currently don't have therapeutic approaches. But that still leaves a large number of CF subjects that won't be amenable to those available modulators. And that's what the second part of this talk is going to focus on. What other therapies and what stage of development they're at for the subjects which aren't at all amenable to therapies now or probably after theratyping is complete. So again, the first approach that we'll talk about is modulators of alternative ion channels. So we're again focusing in on the lung surface. And if you remember, the normal lung, the cilia are happily beaten away, bathed in an appropriate airway surface liquid of a set volume. Now, CFTR is not the only player in terms of defining that ASL volume. Other pathways mediated by other ion channels cooperate with CFTR to set that ASL volume. But of course, if CFTR is dysfunctional, then the ASL is not appropriate. But what about if we target those other processes to try and compensate for loss of CFTR function? If we consider that ASL, its overall volume is set by absorptive processes that suck fluid away from the ASL and secretory processes that add volume to the ASL. So if we inhibit the pro-absorptive processes, we could effectively compensate for loss of CFTR secretion. Or if we activated alternative pro-secretory driving forces, we could again compensate for lack of CFTR function. So here we are modulating alternative ion channels to compensate for loss of CFTR function. In terms of inhibition of pro-absorptive pathways, ENaC has been one protein, the epithelial sodium channel, that's been targeted in this manner. Several companies have tried to block ENaC with small molecules and also with peptides. And unfortunately, to date, none of those drug development campaigns have been successful. But there is still hope that potentially an appropriate ENaC blocker at the right concentration of the right formulation could be effective. Pro-secretory pathways have only been attacked more recently. The reason being because we didn't know the molecular identity of the pro-secretory pathway or alternative pro-secretory pathways in the lung. Turns out that protein was cloned not so long ago. It's called TMEM16A. And Enterprise Therapeutics in the UK have a small molecule which activates that pathway. So that approach hopefully will go into clinical trials in the very near future. So that's the first alternative approach that would be available to all CF subjects because it's agnostic to the CFTR mutation, meaning it's irrelevant what mutation you have. This therapy theoretically should be amenable to you. It's important to take many shots on goal. So another alternative approach that's been used is genetic approaches. And there are two main approaches, which are shown on this slide schematically, that are being used: gene therapy and gene editing. They have similarities, but they're also very distinct. So I'm going to explain the concept of what's going on with these two approaches now. In gene therapy, again, the nucleus contained in the DNA has CFTR with a mutation. Gene therapy just is the process where additional normal CFTR genes are added back to the nucleus. So in no way are we affected by the mutation again. We're just going to add normal CFTR genes into the DNA of the cell. That normal CFTR gene is going to be translated to the normal messenger, which is going to be translated to the normal protein. So again, it's just adding back the right gene. Gene editing is conceptually different in that we focus on the gene that is endogenously in the DNA, and we go in and try and repair that error, the mutation. And that's done with an enzyme, and it's also a targeted approach. So a particular mutation will have to be targeted individually to correct it. That process requires both a molecular guide, which is shown in purple there, but here we're repairing the mutation in the DNA. Both of these areas are highly popular, definitely burgeoning. And on the next slide, explain which companies and which and what level these therapies are. If we first consider gene therapy, there are at least 4 groups attacking this vigorously right now at different stages. 4D Molecular Therapeutics, Spirovan, Abena, and the UK Gene Therapy Consortium are all targeting gene therapy development. What's important here, again, with the concept that multiple shots have been taken at goal, is that these different entities are actually using different types of viruses. So AAV is adeno-associated virus. Spirovan have an AAV program plus a lentivirus program. And for instance, the UK Gene Therapy Consortium have a Sendai virus program. Gene therapy is a little bit further along than gene editing because gene editing is just a newer technology. But there are several companies investigating the utility of this approach and also the adaptability of the approach to actually develop in therapies. Okay, if you remember the first slide, which covered the flow of genetic information, it goes from DNA, the messenger RNA, to the protein. Gene editing and gene therapy are really targeting the DNA. mRNA replacement is the next step along the line. So just replace the mRNA. So we're delivering that intermediary molecule between the DNA and the protein to bypass the problem of the mutation in the gene. In this case, what we need to do is have a carrier, and a lot of the for the messenger RNA, and a lot of the research in this field has actually been targeting development of appropriate carriers for the messenger RNA so that it is delivered to the right cells in a stable form. And again, this is a very active area of research. At least 3 companies listed there on the slide, Translate being the top one, are developing this kind of approach. In fact, Translate Bio are already in phase 1 trials and have broken some of the data for that trial. So there's a great deal of hope in the field that they'll start producing positive data in clinical trials in the very near future in CF subjects. Okay, so what about specific therapies for nonsense mutations or X mutations or premature termination codons, all terms that are interchangeably used for this form of mutation? And again, just to explain the concept, this is a class 1 mutation. So part of the protein actually just isn't generated. So for instance, in the example shown there, the W1282X CFTR mutation, that the protein that's formed there is actually missing the end, so it's non-functional. There's other processes that occur in the cell which go against the production of this protein as well. If you remember again from the first flow of information slide, the process that makes the protein involves the machine called the ribosome, which chugs along the messenger RNA and produces the polypeptide chain. That machine can actually be coaxed to work slightly ineffectively or inappropriately. So instead of actually seeing the premature termination codon in the CFTR protein as a termination codon protein, it's kind of induced and cajoled and persuaded to actually read through that stop mutation and actually produce the full-length CFTR protein. And several efforts are ongoing to find small molecules and to test the feasibility of this approach. And there are various stages. So for instance, the CFFT laboratory has screened over 200,000 molecules, and that campaign has given us a load of tool molecules that are used throughout the field now, and also developed a lot of resources that are used by researchers in the field, such as cell models and cell lines. The Southern Research Institute, in conjunction with the University of Alabama, have screened 500,000 compounds in pretty advanced high-throughput screening campaigns to try and identify molecules. And again, so far, the program is quite nascent. The screening is far advanced, but we'll know, hopefully, in the next few years whether or not any of these molecules will come to fruition. Icogen is another company which is supported by the CFF. They're screening nearly 10 million molecules, both directly and virtually, to try and identify molecules that can enable this process of read-through to occur. And actually in phase 2 trials now, ELOX have a compound which enables this process of the ribosome actually reading through a nonsense mutation to produce full-length protein. So hopefully within the next few months, actually, the phase 2 data will break, and we'll find out whether or not this approach is really working. That's the read-through is not the only approach that's been used for nonsense mutations. At least 2 entities that I'm aware of are considering a slightly nuanced technique to read through the stop codon. So what we're doing in this slide is really focusing down on how the ribosome interacts with the messenger RNA and produces the polypeptide chain, the protein chain, CFTR. Another intermediary molecule is involved in that process, and that molecule is called a transfer RNA. And that really is a true intermediary, because if you imagine that protein as a two-ended entity, at one end it's got the capability to read signals in the messenger RNA, and at the other end of the molecule it's got the ability to select an amino acid and choose the correct amino acid and make that go into the polypeptide chain. A premature termination or stop codon in the wrong place is normally recognized by a transfer RNA which binds to that stop codon but doesn't carry an amino acid into the ribosome. But what we can do is trick that system by engineering, basically mutating the tRNAs so that they recognize the stop at one end, but then also recognize an amino acid at the other end. And then that amino acid is delivered into the growing polypeptide chain. It's a really exciting technique. One commercial entity that Phil's involved with, Therapeutics, is well along in developing this approach for CFTRX mutations. And at least one other academic lab has taken a slightly nuanced approach to it as well. They're making great progress. That's John Lewick's lab in the University of Rochester. So I just want to acknowledge that there's several entities working in this field as well. Okay, just the final slide. We've covered a lot of mutations here and a lot of therapeutic approaches, and I've noticed a lot of the questions are kind of covering this kind of area. Some of these approaches are truly mutation agnostic, which means it doesn't matter what mutation you have. So it doesn't matter what level of understanding we have about what that mutation is doing to the CFTR, because the therapies that are in development are completely agnostic to the mutation. They don't care what the mutation is. because they're targeting some other process. And examples of that, for instance, are the alternative ion channels, such as activation of the TMEM16A prosecretory pathway, gene therapy, or mRNA therapies. All of those approaches, those therapeutic approaches at the various stages of development, they really don't care about what mutation is in the. There are also some mutation-specific approaches. So, for instance, the read-through approach that I spoke about and the tRNA therapies, they're only really in development for X mutations. They're the only mutations that they will be effective on. And gene editing, to a degree, is mutation specific because the editing has to be driven by where the mutation is in the CFTR gene. But it's conceivable that the technology will be such that the editing can be made anywhere, just directed by the different guide molecules. So in some respects, gene editing is mutation specific, but that overall platform should eventually be mutation agnostic. Okay, and that ends the formal section of our presentation.
Right on time. So we're going to head into Q&A. I have been monitoring your questions in Q&A and also in the chat. And I see that a lot of folks have had questions about kind of general clusters of mutations that have particular implications for theratyping and some of the therapies that may be opened up by that approach. So let's start there. And let's talk about some of the emerging frontiers in theratyping informed therapeutics and what we can expect to see in the next few years.
Okay, I guess that question's kind of for me, so I'll just tell you what's happening right now. So just to explain, we're not testing all CF causing variants. We're only testing, so we're not testing nonsense variants. We're only testing variants that make a CFTR protein, but they put the wrong amino acid in some place, or they delete an amino acid in some place. And the reason we're doing that is because those modulator therapies target the CFTR protein. So we limited our analysis to variants that are predicted to make a CFTR protein. So at the present time, we have tested all 650 on the Orkambi combo, and we've identified over 150 of the 650 that respond by the criteria that Vertex used to get approval and label expansion for Orkambi. So that's good news, I think. We have initiated a process with Trikafta, which we hope to be finishing about now, but with the coronavirus shutting the lab down in early March, that's going to get pushed out until who knows exactly where, because we probably have a good 2.5 months worth of work left to do on the Trikafta on these additional variants, but we just can't do the work right now. Does that answer the question?
Pete, do you want to add anything on that topic, or should we move on to another question?
Yeah, I think Phil answered the question very effectively. That's definitely the quickest way that we'll understand whether or not any more mutations are amenable to the therapies we have.
I was going to say, I see a question that asked about theratyping in the clinic that's up. Yeah, so that's actually a different type of theratyping. That's probably based on recovering some cells from, I think it says your son. So recovering some cells and from him and growing those out in a way that the function of CFTR can be monitored either before and then after treatment. So that's really almost more like personalized medicine than trying to put something into a group. It's just like a one-off experiment to see if that particular CFTR gene that your son has produces a protein that would respond to those therapies. And I really can't speak to how this COVID-19 is going to affect those processes, my guess is it's probably not going to have a huge impact on the clinical side collection, but it will have an impact on the laboratory side because, for instance, at my own institution, only essential personnel are allowed into the laboratory. And basically, we're not able to do experiments unless they're related to COVID-19 directly. All we can do is keep our cell lines alive, keep our animals alive, and just do maintenance kind of work right now. That's all we're allowed to do, unfortunately.
It's very similar for us. So,
Yeah.
you know, I grew up in medical school, and my dad is still running research laboratories. He's actually our biomedical sciences chair at the College of Medicine, and he and my mom had a developmental neuroscience lab for many, many years. I had very interesting summers as a child, but right now we are very restricted in what we're able to do that involves direct patient and direct contact. However, as I was saying in the chat, there is a fair amount of flexibility built in with our research pipeline, where even though we may need to adjust the timetable for activities, that's not going to necessarily have a huge impact on the ultimate delivery window for when therapy is going to start to reach. Kind of building on that, we also had a question kind of building on the idea of the fact that theratyping is really two-pronged. There's this personalized aspect, and then there's this group-wise aspect targeting clusters of mutations. We had some folks asking questions in the Q&A about how, when new mutations appear in our registry data, how do we go through the process of understanding what those mutations are doing so that we can get to the point of understanding what they mean for individual and group-wise theratyping?
I'm sorry, I didn't get all of what you said.
No worries. We had folks asking questions about new mutations that emerge in the registry data.
Yeah.
What's the process when new mutations appear for making sure that we understand what those mutations are actually doing so that we can understand what the implications of those mutations are for a theratyping approach to treatment?
Yeah, so in the process that we have going on, the FFT labs, Ross Franklin and UT Southwestern, we're moving those variants as they're identified as quickly as possible into our pipeline as soon as we gain the information from Johns Hopkins and the CFTR2 group. I don't think that's actually germane to the other type of theratyping, which is more the personalized medicine, because the advantage of that is because it looks at the whole gene, because there could be changes elsewhere in the CFTR gene that either makes it a little more active or a little less active, and then you put the CF-causing variant on top. It's more severe, less severe. So testing those cells directly should give you a relevant answer to that specific person, right? And Zan, I see a question about the tRNA and the read-through approaches about actual stop codons. That's actually an extremely savvy question from somebody named Adam Galper. So could I just take a shot at answering that? Because that's a really good question.
Absolutely, let's get into it.
That wasn't my answer, Adam. So I think in the case of the aminoglycosides and the read-through agents that reduce the fidelity of the ribosome, there's no reason to believe that they would have a differential effect on a nonsense mutation, a natural occurring stop codon. The hope is through pharmacology, you might be able to find something that tips that balance. But in fact, the reduction in the fidelity of the ribosome is why those compounds work well as antimicrobial agents. By contrast, Pete did a beautiful job of explaining how the tRNA actually reads the codon, and you can just re-engineer a codon, a tRNA for a specific amino acid. It's a little more nuanced than that, but you could re-engineer it so it reads that nonsense codon as a signal to put in a specific amino acid at that position. Now, why doesn't it do it when you use that stop codon for the natural termination of the protein? That's because there's a lot more to termination than just simply that codon. There's elements of structure in the RNA. There's proteins bound at specific places on that mRNA as it's read by the ribosome. So natural stop codon has a lot of redundancy in what makes it a good stop and effective stop codon. And the nonsense codon doesn't have any of that. It has only the change in the codon. So that's enough to cause disease, but it actually means that it's more of a house of cards. It's easier to tip through and read through that.
And to add to Phil's answer, there's empirically generated experimental evidence that the appropriate stop codons, the real stop codons, aren't being read through. Yeah. And that's work that, it's a really important question. The CFFT are really investigating that. In fact, Bill Scatch has driven that effort.
All right. And we have some questions in the Q&A about whether we can generalize at all about rare mutations and their comparative impact on disease presentation and severity versus some of the more common mutations like DF508.
I think that's a tough question. Even if we look at the knowledge base that we have about F508del, we know that the presentations of disease are variable in those subjects. So extrapolating any knowledge we have from there, we're already looking at a broad spectrum of disease. Almost every year I have a colleague that tells me some fantastic story about a 70-year-old that they've just diagnosed with cystic fibrosis that's F508del. So there's a broad spectrum underlying this disease in terms of presentation. So it's difficult to predict, I think is the right answer.
So my doctor actually had one of those cases when he was a medical resident. Somebody who had never had any symptoms before she was in her late 60s, double delta 508. And I'm sitting there like, yeah. So everybody's very different. And that really speaks to some of the other questions that we've gotten as well about how different mutations can kind of interact with each other, how those consequences vary according to a variety of circumstances. And as a sociologist who has a biomedical background, something I can comment on also is the fact that it's not all about the genome, which is basically the switchboard that you start out with in life, with switches that can be turned off and on. It's also about the epigenome, which is basically the program that tells the switches to turn on and off and how much on and how much off and when. So you start out with a particular set of generic characteristics, but those are not the be-all and end-all of what actually happens. So what we see with a lot of the folks who are the double deltas, who are what we consider to be long-term non-progressors, are that there are things that are either buffering against the negative impacts of those mutations on the ability of salts to move across the cell membranes, or that there are things happening elsewhere in their genome that are actually suppressing the activity of those mutations themselves. So no two of us are alike, even if we have the same mutations. And then we have questions about genetic modifiers versus the CFTR protein modulators. Well, that's sort of another angle on that, is that we have to understand first and foremost, what are the unique impacts of the actual genetic mutations and the epigenetic activation of those mutations that a person is starting out with? What does that mean for personalized medicine with that individual? We have maybe one more minute for Q&A before we got to transition into the final housekeeping. Let me just take a quick peek at our tab and let me make sure that we kind of hit the biggest items. Let me wrap up with just this one question about mRNA therapies. We've had a lot of activity about those in the chat as well. Do y'all reckon that mRNA-specific therapies are probably our fastest route to something that resembles a mutation-agnostic cure?
Absolutely. because you're going to just replace, rather than the CFTR normal gene, you're going to just kind of short circuit the system and replace the normal CFTR mRNA. The difference is that it's not a permanent thing, because the mRNA gets made by the DNA and it only lasts some finite amount of time. So that's why Bill was saying in his plenary lecture that this is going to require repeat treatments, where every couple of weeks you may have to get another treatment. By contrast, with the gene therapy or the gene editing, if you can hit an adult stem cell that's the genesis of all the new epithelial cells, one could hope that a one-shot treatment might persist. But it is mutation agnostic. The mRNA, you're just putting the correct CFTR back in on top. Yeah.
Super. Pete, anything to add before we wrap up? All right, so I'm just going to do a little bit of final housekeeping here before we close out. First and foremost, I want to direct everybody to the resource room. We've got lots of goodies in there for y'all, including copies of the slides, a glossary, and a few other materials that'll help you dig deeper into the science. We've also got our contact information in there. I do encourage you to follow up with us if you have additional questions, or maybe you're looking for pointers on how to get involved in some next-gen research. Finally, we also want, for those of you who came in late, to encourage you to connect with the CF Foundation's Community Voice Initiative. You can do that by emailing our CFF staff or go on the website. That's another great way to get involved in research and give your feedback on a huge variety of topics within genetic specific therapies for rare nonsense mutations. So with that, thank you all very much for coming to our session. This has been fantastic. We hope we did a good job of answering your questions in the limited time we've had. I want to give a big thank you to Pete and Phil for their outstanding contributions, as well as Genevieve Mall from CFF, who was instrumental in putting together our slides. Kelly Stavroulakis, who was our support from CFF in making sure that we could get this session off without a hitch. With that, I'm going to encourage y'all to take a bio break, maybe get a snack, do your airway clearance, whatever pleases you. Take 15 minutes just to kind of reorient, and then return to the general event environment for the next breakout session that you signed up for. And if you're having technical difficulties, please hit that support tab to make sure that you can have a good experience in your next content module. We hope everybody else enjoys the rest of the conference. This has been great. Thank you all so much for coming. Phil, you're on mute.